In the last class, I have explained about enzymes in genetic engineering. So far you have seen DNA polymerases and nucleases. Today, I would like to explain about ligases. DNA ligases. While exact cutting of DNA molecule is very useful for DNA cloning, its full potential is only exhibited when the fragments produced are joined together to give a new structure, known as recombinant DNA. This joining or ligation is achieved by the use of a DNA ligase enzyme. The most common ligase enzyme is isolated from the T4 bacteriophase. Along with the T4 bacteriophase, we have DNA ligase also. The DNA ligase forms a group of enzymes which mediate annealing, sealing, or joining of DNA fragments. Primarily, ligase enzymes are involved in the repair of DNA molecule, where sealing or union of DNA fragments takes place. DNA ligases also play active part in processes such as DNA replication and recombination. These enzymes are widely used in genetic engineering for the production of hybrid DNA. Since ligase enzymes join DNA fragments or seal the nicks in the chain, they are called molecular scissors. Activity of ligase enzymes. Ligation of DNA molecules with sticky or cohesive ends. If two different DNA preparations are treated with the same restriction enzyme to give fragments with sticky ends, these ends will be identical in both preparations. Thus, when the two sets of DNA fragments are mixed, base pairing between sticky ends will result in the coming together of fragments which were derived from different molecules. Also, there will be pairing of fragments derived from the same molecule. Such pairing are temporary owing to the weakness of hydrogen bonding between the few bases in the sticky ends. The pairing can be stabilized by the use of DNA ligase, which forms a covalent bond between the 5' phosphoryl at the end of one strand and 3' phosphoryl of the adjacent strand. Polynucleotide ligase enzyme of T4 bacteriophase catalyzes the end-to-end joining of DNA duplexes at the base paired end. This reaction could occur intermolecularly or intramolecularly. Researchers confirm that intermolecular mode of reaction is correct. The ligation reaction is derived by ATP and it's carried out at 4 degrees centigrade to lower the kinetic energy of molecules. This reduces separation of paired sticky ends and are later stabilized by ligation. However, long reaction time is required to compensate for the low activity of DNA ligase enzyme in the cold. The enzyme concentration is kept high and polyethylene glycol is added to reaction mixer for stimulation. Since ligation reconstructs the site of cleavage, recombinant DNA molecules produced by ligation of sticky ends can be cleaved again at the joints using the same restriction endonuclease enzymes that was used to generate the fragments initially. As a result, fragment can be inserted into a vector DNA and recovered again after cloning of the recombinant molecules. In this picture, you can see the mode of action of ligase enzyme during ligation of DNA molecules with cohesive or sticky ends, complementary cohesive ends base pair forming a temporary link between two DNA fragments. This association of fragments is stabilized by the formation of 3' to 5' phosphodiester linkage between cohesive ends, a reaction catalyzed by DNA ligase. Next, ligation of DNA molecules with blunt ends. Fragments of blunt-ended DNA can be ligated, but since there is no base pairing to hold fragments together temporarily, concentrations of DNA and ligase enzyme must be high. However, blunt end ligation is a useful way of joining together DNA fragments which have not been produced by the same restriction enzyme and which therefore have mismatched sticky ends. These ends are removed prior to ligation using the enzyme S1 nuclease, which digests single-stranded DNA. In this picture, you can observe the mode of production and ligation of blunt ends. Different blunt-ended DNA fragments cannot be held together temporarily by base pairing between cohesive ends, and so both DNA and ligase must be used at high concentrations to increase the enhances of two DNA fragments occupying the ligase active site simultaneously. In case of ligation of blunt ends, the restriction site will not be In case of ligation of blunt ends, a restriction site will not be regenerated and thus may prevent recovery of a fragment after cloning. For this reason, short DNA duplexes called linkers are frequently used for joining DNA. Linkers are short double-stranded oligonucleotides with blunt ends containing at least one restriction site, that is EcoR1 palindrome, within their sequence. These linkers can be joined to one preparation of DNA by blunt-ended ligation, and then sticky ends can be created by cleavage of the linkers with a suitable restriction enzyme. The linker is chosen so that the sticky end it produces is identical to that on the other DNA preparation. Consequently, the two can then be joined by ligation of their sticky ends. Some very versatile linkers are available which contain restriction sites for several different enzymes within a sequence of only 8 to 10 nucleotides. Blunt-ended molecules can also be ligated after building sticky ends. Thus, with this method, it is now possible to insert a foreign DNA segment at a particular site in the linker region of the vector and then retrieve this foreign DNA segment whenever necessary. Sources of DNA ligases. There are three types of DNA ligases are there. They are E. coli DNA ligase and bacteriophage T4 DNA ligase and T4 RNA ligase. DNA ligases are isolated from E. coli. The ligase enzyme is isolated from E. coli is a polypeptide chain with a molecular weight of 74 kilodaltons. It requires NAD plus as cofactor. The ligase this enzyme has narrower substrate specificity, making it useful tool in specific application. Back to refers T4DNA ligase. The ligase obtained from T4 bacteriophage is 68 kilodalton. The most widely used T4 DNA ligase is derived from the T4 bacteriophage. It is a monomeric polypeptide. The molecular weight 68 kilodalton is encoded by bacteriophage gene 30. It has border specificity and repairs single-stranded nicks in duplex DNA, RNA, or DNA RNA hybrid. It requires ATP as a cofactor and a source of energy. For the routine laboratory requirements, T4 DNA ligase is obtained from an induced lysogen of lambda T4 LIG phase. This enzyme has the capacity to ligate a variety of cohesive and blunt-ended DNA fragments. The enzyme concentration is kept higher and a fucogen called polyethylene glycol is added to reaction mixture for stimulation. The third type of T4 ligase is T4 RNA ligase. T4 RNA ligase is the only phase RNA ligase that has been extensively characterized and used in genetic engineering. This enzyme catalyzed the phosphodiester bond formation of RNA molecule with hydrolysis of ATP to PPI, proton pump inhibitor. It is monomeric enzyme with 373 deduced amino acid residues is a product of the T4 gene 63. Applications of DNA ligase enzymes. DNA ligase enzymes play an important role in genetic engineering. In the absence of DNA ligase enzymes, recombinant DNA technology cannot be successful. The important functions of DNA ligase are genetic engineering experiments involve joining of DNA fragments to produce recombinant DNA molecules. Ligase enzymes are used in the joining processes. Ligase enzymes help in ligation of vector and inserting recombinant DNA. They help in ligation of linkers or adapter molecules at the blunt ends of DNA fragments. They help in sealing nicks in double-stranded DNA. Ligase enzymes require 3' hydroxy and 5' phosphate group for ligation. This requirement can be advantageous as self-ligation of DNA can be prevented by dephosphorylation using alkaline phosphatase of intended donor fragments prior to ligation. Next, dephosphorylation of vector DNA will prevent recirculation of the vector in cloning procedure. The summary of DNA ligase. DNA ligase catalyzes the formation of phosphodiester bond between the 5' phosphate of one strand of DNA or RNA and the 3' hydroxyl of another. ATP is required for the ligase reaction, which proceeds in 3 steps. One is adenylation, that is addition of AMP of a residue in the active center of the enzyme. Phosphoryl and second, transfer of the AMP to the 5' phosphate, so-called donor, formation of a pyrophosphate bond. Third one, formation of a phosphodiester bond between the 5' phosphate of the donor and the 3' hydroxyl of the receptor acceptor. Depending up Depending upon the source, the enzyme requires either ATP or NAD plus as cofactors. E. coli DNA ligase, it is derived from E. coli cell and requires NAD plus as cofactor. T4 DNA ligase requires ATP as cofactor. These are the silent features of DNA ligases. Next enzyme, terminal deoxynucleotidal transferase. The enzyme deoxynucleotidal transferase catalyzes the repetitive addition of monodeoxynucleotide units from a deoxynucleoside triphosphate to the terminal 3' hydroxyl group of a DNA molecule. This enzyme has a molecular weight of 32,000 and consists of two subunits, each with a molecular weight of 26,500 and a 80,000 unit. Enzyme is isolated from calf thymus. Terminal deoxynucleotidal transferase is a template independent DNA polymerase because it is able to synthesize a new DNA polynucleotide without base pairing of the incoming nucleotides to an existing strand of DNA. Terminal deoxynucleotidal transferase is expressed mostly in the primarily lymphoid organs like the thymus and bone marrow. This enzyme catalyzes the addition of nucleotides to the 3' terminus of a DNA molecule. Cobalt is a necessary cofactor. However, the enzyme catalyzes reaction upon magnesium and manganese administration in vitro. This enzyme is used for the formation of a cohesive and homopolymer tailing. A homopolymer is simply a polymer in which all the subunits are the same. Enzyme terminal deoxynucleotidal transferase catalyzes the addition of a series of nucleotides on the 3' OH terminal of a double-stranded DNA molecule. Use of terminal transferase enzyme. The enzyme terminal transferase is used to add homopolymer tails of DNA fragments using a technique called homopolymer tailing. Sticky ends can be built up on blunt-ended DNA molecules. For examples, one preparation of DNA could be treated with the enzyme terminal transferase. In the process, one preparation of DNA could be treated with the enzyme terminal transferase in the presence of DATP, resulting in the addition of a poly chain to the 3' end of each DNA strand. There is another preparation of DNA which provides 3' tails of poly using same enzyme with TTP. When both types of DNA preparations, that is DNA fragments with poly A tails and DNA fragments with poly T tails, are mixed, there takes place base pairing between complementary sticky ends, which could then be ligated. One advantage of this method is that ligation does not take place between fragments from the same DNA preparation. In this picture, you can see the method of homopolymer tiling by terminal transferase enzyme. One DNA preparation is given by poly tiles at its 3' end. and the other receives polytiles. These tiles tend to hybridize with each other when the two preparations are mixed, and ligation can be used to stabilize this association. If the tiles are long enough, hybridization tends to be so stable that ligation is not required. Other uses of terminal transferase enzyme. Terminal transferase enzyme is used for the 3' and labeling of DNA fragments, and terminal transferase enzyme is also used for the addition of single nucleotides to the 3' end of DNA for in vitro mutagenesis. Purine and pyrimidine polymerization rates by using terminal transferase enzyme depends on the addition of magnesium 2 plus, manganese 2 plus, or cobalt 2 plus in the reaction mixer. The next enzyme used in genetic engineering is alkaline phosphatase. The enzyme alkaline phosphatase catalyzes the removal of the 5' terminal phosphate residues from nucleic acids RNA, DNA, and ribo and deoxyribonucleotide triphosphates. This enzyme is isolated from bacteria or calf intestine. This enzyme is a dimeric glycoprotein which a molecular weight 14,000. It is made up of two identical or similar subunits, each with a molecular weight of 6,900. It is a zinc containing enzyme with 4 atoms of zinc 2 plus per molecule. Functions of alkaline phosphatase enzyme in genetic engineering. Linearized cloning vectors can be prevented from recirculating by dephosphorylation with alkaline phosphate enzyme. The free 5' hydroxyl group can be phosphorylated with polynucleotide kinase and gamma P32 ATP to produce 32P and labeled nucleic acid. Alkaline phosphatase enzyme is used for mapping and DNA fingerprinting studies. Summary of alkaline phosphatase. Alkaline phosphatase is purified from either E. coli or higher organisms. It is used for removal of 5' phosphate groups from nucleic acids in order to prevent recirculation of DNA vectors and cloning experiments. Although E. coli and Although E. coli and calf intestine alkaline phosphatases have different structures, both enzymes are zinc and magnesium containing protein. Bacterial alkaline phosphatase is more stable but less active than calf intestinal alkaline phosphatase. The next enzyme used in genetic engineering is polynucleotide kinase. This polynucleotide kinase catalyzes the transfer of the terminal phosphate group of ATP to the 5' hydroxylated terminal of DNA or RNA. Polynucleotide kinase enzyme is frequently used to unlabel the nucleic acid with P32. This can be accomplished by forward reaction or exchange reaction. In forward reaction, transfer of labeled gamma phosphate form ATP to the free 5' hydroxyl group of the substrate. Substrate lacking a free 5' hydroxyl requires prior dephosphorylation by alkaline phosphatase. Second reaction is exchange reaction. In the terminal step, the terminal 5' phosphate is transferred from substrate to ADP present in the reaction mixture. Then the labeled gamma phosphate from ATP complex is transferred to free hydroxyl group of substrate. Polynucleotide kinase enzyme is used to label 5' terminal of DNA and RNA with gamma p32 ATP by phosphorylation of 5' hydroxyl groups or by the exchange reaction. This 5' terminal labeling is used in mapping of restriction sites, DNA or RNA fingerprinting, hybridization studies, and sequence analysis of DNA. Polynucleotide kinase summary. Polynucleotide kinase, the T4 polynucleotide kinase obtained from E. coli cells infected with T4 phase. It performs the reverse reaction of alkaline phosphatase. It is adding phosphates to 5' ends and mainly used for end labeling of DNA molecules. It catalyzes the transfer of the gamma phosphate from ATP to the 5' OH groups of single and double stranded DNAs and RNAs. Oligonucleotides or nucleosides, 3 monophosphates forward reaction. The reaction is reversible. In the presence of ADP, T4 polynucleotide kinase exhibits 5' phosphatase activity and catalyzes the exchange of phosphate groups between a 5' P oligo polynucleotides and ATP. This is called exchange reaction. Next enzyme used in genetic engineering is S1 nuclease. The S1 nuclease enzyme is a single strand specific endonuclease which cleaves DNA to release 5' mono and 5' oligonucleotides. Normally, double stranded DNA, double stranded RNA, and DNA RNA hybrids are resistant to action of S1 nuclease enzyme. However, very large amounts of S1 nuclease enzyme can be completely hydrolyzed double-stranded nucleic acids. The enzyme hydrolyzes single-stranded regions in the duplex DNA such as loops and gaps. S1 nuclease enzyme can also cleave single-stranded areas of superhelical DNA at torsional stress points where DNA may be unpaired or weakly hydrogen bonded. Once the superhelical DNA is nicked, S1 nuclease enzyme can cleave the second strand near the nick to generate linear DNA. S1 nuclease enzyme is a monomeric protein with 3800 Dalton molecular weight. It requires zinc 2 plus for its activity and is relatively stable against denaturing reagents such as urea, SDS, and formamide. The optimum pH requirements lies between 4 to 4.5 for S1 nuclease enzyme. The applications of S1 nuclease enzyme. S1 nuclease enzyme is used to analyze DNA RNA hybrid structures to map transcripts. It can be used to remove single stranded tails from DNA fragments to produce blunt ends. Hairpin loop structures formed during synthesis of double-stranded complementary DNA is digested by this S1 nuclease enzyme. And S1 nuclease enzyme is also used for DNA mapping called S1 nuclease mapping. S1 nuclease enzyme was extracted from Aspergillus oryzae. The S1 nuclease is a specific single-stranded endonuclease. It can degrade single-stranded DNA and single-stranded RNA to produce 5' single-stranded nucleotides and oligonucleotides. Double-stranded DNA and DNA-RNA hybrid molecules are more resistant to S1 nucleases, and only high concentrations of enzymes allow them to be digested. And I already told you the optimum pH for the enzyme reaction was 4 to 4.5. This enzyme requires low levels of zinc 2 plus activation. Some chelating agents such as EDTA and citric acid can be strongly inhibits S1 nuclease activity. In addition, phosphate buffer and 0.6% SDS solution can inhibit its activity, but it is in addition, phosphate buffers and 0.6% SDS solution can inhibit its activity, but it is stable to urea and formamide. Its activity is similar to Mung-Bean nuclease. However, the enzyme will also cleave a strand opposite a nick on the complementary strand.